ASRB NET 2024, just especially related to plant pathology. So the application is not released yet, but probably we're expecting very soon, probably maybe in the second week or maybe third week or at the end of this August month, as far the sources are saying. So most probably we'll get at the end of this month, and we may get maybe 2 months or maybe 3 months or like the time in between the application and as well as exam notification and as well as exam. So most probably in the last time the net gave only 40 days or 40 day or 42 days, I think. I'm not sure exactly, in between 40 to 45 days. The time is very less, so we cannot expect maybe this time also they conduct like that only. So but I believe that at least we will get at least 2 months time, so that you can prepare well regarding net. So let me tell you, like how to prepare yourself. There are lots of mails we are getting, many people are using asking like how to prepare. Not, we are getting worried this time, we need to get the net done and all those things. So rather than asking someone else, in fact you guys also even you guys, not only you, anybody, no need to worry about it. You can prepare yourself without anybody's help, include our help. And in this we're going to tell how to prepare yourself, like what are all the things that you need to keep it in your mind, and like what are the things that we must concentrate to make our to get the net done this year. So I'm N.S. Shankar Reddy, PhD plant pathology. So we'll start with how to prepare yourself. So first of all, how to prepare. So everyone have a doubt how to prepare, where to start, what all the books need to study, all those things. So actually at the end of this presentation you will get how to prepare yourself. You will get an idea where to start and what to do the things. The second thing, what are all the areas we must concentrate, like mycology or bacteriology or virology. There are different things will be there, right? So what are all the must concentrated areas? What are all the things that we must take into consideration? So what are all the areas to concentrate? The second thing we're going to discuss. The third thing is method of preparation, how to prepare. So reading the books or taking the hints, no, like everyone have their own preparation method, but I will tell you what might be or what will be the best method for the preparation. The next one is the making notes, how to make notes. So we have a very short time, so that in the short time how to make notes and how to recall all the things in a very short span of time, that we will tell. And how to read books. So there are plenty of books are related to, especially related to pathology. There are n number of books in market that may be related to MCQs, that may be related to the preparation of net, that may be related to preparation of ARS, and various other kinds of books are available in the market. So what are all the books that we need to choose, and what are all the basis of choosing, like how to read the books and all those things that we will discuss. And the next thing, what are all the books to be preferred? And there are plenty of books that I told you. N number of books are there. That if you see, all the books more or less contains, almost all kinds of books contains more or less similar kind of information only. But we will select some couple of important books, like what are all the books to be preferred to have a better knowledge. The second thing. Next thing is repeatedly asked question is many people are asking is GN Agrios book is necessary for the preparation of net alone? For only preparing net, is GN Agrios book is necessary? That we will discuss. Next thing is the previous year net question paper. What will be the impact of previous year net question paper? Why do we need to study previous year net question papers? And next, what to do in the last 1 week before the exam. So everyone were a little bit anxiety or some kind of in a dilation mind, like what to do in the last 1 week and how to prepare. And so so many things will be there, right? So I'll tell you what to do in the last 1 week before the exam. And the most important thing is, we will clarify you the doubts, whatever the doubts you have regarding to preparation, books, everything. We will help you. The geek researchers are there to help you. There is no need to worry about for 2024 preparation. Not only 2024 preparation, we have already published one video regarding that. We are giving free training or free kind of things for not only net, but also JRF, but also ARF or ARS and all the other kinds of things. We are giving almost free services, like all the students, whoever you are, even though if you are planning from BSc to MSc through for plant pathology, like that is JRF generally, and to have a broad spectrum knowledge also, we can train you the students for free of cost. We'll start with the first thing, how to prepare yourself. This is the most commonly asked question, sir. How to prepare, sir? What are the things need to consider, sir? How to prepare? So the preparation strategy may be different from people to people, because some people, just read the facts, or maybe someone will take the notes, or somebody will do different kinds of strategies will be there, because individuals are different, so that mindsets also will be different, so that the preparation strategy will be different. But at the end of this, you will come to know how to prepare yourself. You will have an idea that what are all the things you must keep it in your mind. That kind of things will tell you. What are all the areas to concentrate? The first question. Like if you see the pathology, it's not that small, right? Like mycology, bacteriology, virology, epidemiology, so many things are there. What are all the areas to concentrate? What are all the aspects to must take into consideration? With having this idea in our mind, imagine we prepared some couple of things, like just imagine if you have only 2 months to prepare, or maybe if you have only 30 or 40 days to prepare. So this schedule will be helpful. Even though if you have 50 or 60 days also, this schedule will be helpful. The areas we must concentrate, we have figured it out. The first thing, the first 1 and 2 days we will concentrate on the history. Then you guys will ask me, why is 2 days for history? On the left side, this is serial number, and that is one, this is the days, how many days it will be required to concentrate on this. You guys can ask me, why 2 days for history? See, just imagine how vast the history is, like mycology, bacteriology, virology, epidemiology, applied plant pathology, mushrooms, whatnot. There's a lot of things which was there in the history. And if you recall the last 10 years, net question papers, and we have already published last 10 years net question papers, memory based question papers from 2006, 9, 14, 14, 1, 14 to 18, 1, 18 to 19, 21, 23, and all those things we have published. You guys just take a look into. Every year at least 5 to 10 questions will be from history, that to normal kind of questions. We have sorted out that history is a very important one. We can definitely get 8 to some in between 5 to 10 questions, that to normal kind of questions. That's why I told you to concentrate on the history for 2 days. Make sure that you can no point is like we can leave this point or we can study that point. There is nothing like that. You must go through every single point, mostly like books. What are all the books written by that terms coined by, like contributions and all those things, like books and contribution, all the other things, like like some kind of names, or like life cycles, various kinds of things will be there in the history. So regarding history, we have published a n number of videos. Anyway, this year we will do a history video again. There is nothing to worry about it. We have sorted out a net history of mycology, history of bacteriology, virology, and other things, whatnot. Everything we have prepared a video, so that will be helpful for you. Anyway, you concentrate on the next, since the areas we must concentrate history. So important areas, mycology, bacteriology, and virology, we must concentrate. Of course, other areas also very important. Coming to the introduction to mycology, so general characters, like you don't need to study point by point, point by point. Imagine, no, you just consider that you are preparing for an exam. What kind of exam? Net exam. They will give you MCQ kind of questions. So you must read the points. You don't need to remember. I cannot say you can't remember. Like you just go through the points so that you will have a general idea. Like what are the modifications, specialized structure. Very important thing is the examples of each and everything is important. The second one, third one is, fourth one is fourth day you concentrate on reproduction. And reproduction in fungi is two types: sexual reproduction and asexual reproduction. And the types of flagella, sexual and asexual spores that is produced by fungi, you can concentrate. Like for example, you can consider sexual reproduction. Like we can expect questions like, what are all the kind of question in sexual reproduction? There are three types of sexual reproduction: plasmogamy, karyogamy, and meiosis. In plasmogamy, again there are 5 types, like gametogamy, gametangiogamy, gametangy, spermatization, somatogamy. So they will ask questions like Pythium and Phytophthora can reproduce. What kind of sexual mechanism is involved in Pythium and Phytophthora? Generally, if we study gametangiogamy, where male gametes is called antheridium, female is called oogonium. I'm just telling you the examples, like what are all the areas we concentrate. Nothing to worry about it. We will help you and we will train you. Nothing to worry about it. So like gametogamy, male and female are coming in contact and fertilization tube will be developed. That is called gametangial contact type. Such kind of reproduction can happen in Pythium and Phytophthora examples. So in such a way also we can expect the questions that I am generally telling. You can read in that way. Types of flagella, they will give examples like Plasmodium poromycota can produce which kind of flagella, or else they will tell you the flagella like anterior tinsel and posterior whiplash type of flagella is produced by. And if we dwell deep into that, that contains hair, tin cell, without hair, whiplash, and where this energy coming from, and what is the attachment point of flagella called blepharoblast, various other kinds of things will be there. You can read in that way, and we'll also tell you the books that is preferred. No need to worry about it. And coming to the fruiting bodies, only two types will be there: sexual and asexual fruiting bodies, right? You can concentrate on that first thing. And the next, when it comes to the taxonomy rules and regulations, who is the father of taxonomy, and what all the regulations will be there, what is binomial, what is trinomial, and all the other things. Like most important thing is like a classification system, like two kingdom classification was given by whom? Three kingdom, four kingdom, five kingdom, Whittaker, very very important. And five kingdom classification was by Whittaker. In such a way you can prepare, like it is not for preparing of mid semester, it is not for preparing of semester exams, it is not for preparing of some other things. Net means you can prepare like a net. I will tell you how to prepare. You just remember the points, like key points, you can note it down. And the one more important thing I would like to tell you is put a note, very big note, take the points, every single point, not necessarily needed to take the every single point, but important point you must remember. Important point you must note it down on the notes. I will tell you what will be the benefit of this writing down the points or important points in the notes. I will tell you at the end. Okay, then it's better to go with the present classification. Of course, there is one more classification in 2021 also, but we are mostly following this Kirketal classification that was published, given by Kirketal in 2008. So through, by considering the Kirketal classification, on the 7th day you can concentrate on Kirketal classification, especially protozoa and myxomycota. And mostly this protozoa, clubroot of cabbage, myxomycota, some other kind of fungi, saprolignin, all those things will be there, right? Saprolignin, that is also different, right? So all those fungi will come under their special features, very very important features of the fungi, special characters of the fungi. For example, I will tell you what is the special character of Plasmodium. If you ask me, it will produce a cyst. There is no sexual spore. Asexual spore, juice spore, we know first thing. And the production of a Plasmodium, it will produce no hyphae, no all those things. Production of Plasmodium is a special feature of the life cycle of what? Only Plasmodium poromycota group of fungi produces production of Plasmodium. Like in a such a way, I'm telling, like you can prepare in that way. Okay, in myxomycota also. Myxomycota, for example, you can take saprolignia. What is the special feature of saprolignia? You ask me. Saprolignia, there is one speciality, even though it comes under no. It contains two kinds of zoospores. Like probably, I'm sorry, this saprolignia actually comes under oomycota, I think so. I'm not not myxomycota, sorry for that. This saprolignia comes under oomycota generally. Anyway, since the time came, I will tell you the next classification is like oomycota, pythium, phytophthora, and downy mildews, and also saprolignia you can consider. For example, I will tell you, so all oomycota consist of cellulose and hydroxyproline as their cell wall, but there are a couple of fungi, even though they comes under oomycota, they contains chitin in their cell wall. So such kind of fungi is known as chitin containing oomycota group of fungi. For example, Achaila, Aphadogyla, Saprolignia, Leptometus. And this Saprolignia contains one more special feature, like production of two kinds of zoospores. That is called dimorphism, or we can also call it as diplanetism. Probably the diplanetism is the exact terminology that is required. Like such kind of things you must concentrate. Okay, and just remembering the important points is a very very important one. Next, the classification in the 9th and the 10th day you concentrate on the diseases caused by Pythium, Phytophthora, and downy mildew genera. In fact, I would say in the 8th day itself you can finish this all those things. Anyway, I have given 3 days to this, but in the first day itself you can finish all those things. Anyway, we will tell you classification. Next one is in the 11th day Chytridiomycota, 12th day Zygomycota. And the 13th day classification regarding Ascomycota and general characters of Ascomycota. There are various general characters are like how Asca is formed, ascosporogenesis, we can say, like general characters, asexual fruiting bodies that is produced by hymenium, like sterile hair-like structures, and all those things we can along with examples based on their sterile hair-like structure, how it is growing, such kind of things also we can observe in Ascomycota, right? So such kind of things only we do expect in the net. Okay. And the 14th day Ascomycota powdery mildew genera. It's a powdery mildew genera, little bit big. I cannot say too big, it's a little bit big. So like clistothecial appendages. Types of powdery mildew genera, clistothecial appendages, sericinoid or coil kind of appendage, various kind of things we do study in, you can study in the 14th day. And the 15th day concentrate on anamorphic spores, anamorphic fungi. There are plenty of anamorphic fungi there, right? 16th, 17th and 19th day, what are all the concepts left on the Ascomycota? There are various things are there like Aspergillus, Penicillium, and small kind of mushrooms, like we can say truffles, many other kinds of some kind of medicinal or costly kind of mushrooms are actually comes under this Ascomycota, but majority of the mushrooms actually comes under Basidiomycota, but couple of fungi like as I told you, a couple of fungi that comes under Ascomycota as well, like along Pezizomycotina, I would say Pezizomycotina, right? So that is one of the crown group of fungi of Ascomycota. We will study, like on the 16th, 17th, and 19th day, what are all the topics that left on the Ascomycota we'll study. The next one, on the 20th day, we'll study about Basidiomycota, we'll study about Agaricomycota, especially in mushrooms. Okay, general characters of Basidiomycota along with the Agaricomycota, like Agaricus, Boletus, Manita, various kinds of mushrooms comes under Agaricomycota. Okay, and the 21st day we are studying about Puccinio-mycotina, that is the rust and rust generation disease caused by them. And 22nd day we are going to study about Basidiomycota, Ishtolagnomycota, Mycotina, smart genera and diseases caused by them. And 23rd day you concentrate on Basidiomycota leftover points. Is there is anything left over? So such kind of things you can concentrate on. The 24th day you can concentrate on mycorrhiza, lichens. There are any leftover points. It's not that much important. I cannot say the questions cannot from this mycorrhiza. And there is one commonly asked question is like hyphal nets or some kind of hairs will be present in ectomycorrhiza, mycorrhiza, something will be there. Actually forgot sorry for that. Anyway, in discussion we might have recall these points. Anyway, on the after 20 day, I suggest you to just go through the overall review of mycology. So just so far in the last 20 days you might have studied the various things of mycology. I suggest you to go the overall review of the mycology. That is a very, very important one. On the 25th day, you just started, starting a list of important plant pathogenic diseases caused by them. For example, Agrobacterium, Irvinia, Pseudomonas, Xanthomonas, and various kinds of important plant pathogenic bacteria with there, right? So just list out the important plant pathogenic bacteria and what are all the diseases caused by them. Is there is any special characters and the special media recommended? For example, I will tell you, for example, Pseudomonas, Kingsby, Xanthomonas, it produces yellow color pigment. Such kind of, if there is any specific points that the streptomycin, it contains, antibiotic, right? So such kind of things you concentrate on. On the 26th day, concentrate on cell structure and components, like what is plasmid, what will be the use of plasmid, what is flagella, what are the different kinds of flagella will be there along with the examples. The 27th day, concentrate on recombination and staining of bacteria. And of course, I would say on the 27th day, you can guys are concentrate on growth curves, pigmentation produced by the bacteria, and antibiotics also produced by the bacteria. 28th day, concentration on classification of important antibiotics, mode of entry of plant pathogens, like some pathogens enter through hydrodes, some pathogen enters through stomata, some pathogens through trichomes. Various kinds of entry points are there, and these entry points are very, very, very important. You just take a look into the last 10 years question paper. At least there will be a question from these entry points. So we can concentrate on the common areas where we can get at least 1 or 2 questions, right? At least putting all these 1 or 2 questions, so that will be a great number at the end. Even small facts matters, man, right? Okay, anyway, jokes apart. Selective medias, what are all the different kinds of selective medias are recommended. On the 29th day, concentrate on FEV, fastidious vascular bacteria, endospores, types of secretion system, and along with the survival and spread of the plant pathogen. On the 13th day, concentrate on phytotoxins that is produced by the plant pathogenic bacteria, and the list of important plant pathogenic bacteria that causes diseases, and vector-borne plant bacterial disease. Generally, we know that viruses are known to transmit to plant viral disease, right? The 2 vectors, and there are a couple of bacteria that can also transmit by mediating the insect as a vectors. So we will see what all the different kinds of things will be there. On the 30th day, so have a overall review about bacteriology and as well as mycology, because you have already read the mycology, right, in such a way. Okay. The next one, on the 31st day, concentrate on the general properties of plant viruses, morphology, viroids, viroids, and what are the different types of viroids, like classifications and all those things, virusoid, proteins, or satellites, like different groups of plant pathogens that actually grouped under this virology or viruses, right? And the 32nd day, concentrate on mycophages, lambdaphages, cyanophages, pytoplasmas, pyroplasmal, and bacteriophages, and the list of important diseases caused by them, and their important special features. Every single thing you must concentrate on the special features along with the examples. And 33rd day, concentrate on tobacco mosaic virus, mechanism of infection. 34th day, and you can do the same day, right? On the 33rd day, you can concentrate on TMV, mechanism of action, and transmission of plant virus. Everything puts together in a single day. Okay. On the 34th day, transmission of plant viruses, and the 33, 34 and 35, 36, 37, I've suggest you if there is any leftover points which was there in the virology along with the overall review about virology, bacteriology and mycology. That's why I give 3 days, because very few leftover concepts might be there virology if you studied all those things. After that you can also concentrate on review of overall the things like mycology, bacteriology, overall things that what you guys have done so far. 37th day, I suggest you to concentrate on epidemiology, disease triangles, factors affecting the disease triangle, epidemics and types. 38, pathogenesis or disease cycle, defense mechanism and host pathogen interaction. On the next day, remote sensing, computerized models, gene to gene hypothesis, survival and spread of plant pathogens. 40th day, remaining leftover concept if there in the epidemiology, you can just concentrate on that. The 41, fungicides, classification of fungicides along with examples, very important. Classification of fungicides, chemical nature, based on the chemical nature, seed treatment methods and application of seed treatment method, special methods of application of fungicides, trade names of the fungicides. 42nd day, concentrate on antifungal and antibacterial antibiotics, mode of action of fungicides, mycoherbicides and IPM. And you can take one extra day like considering a quarantine. What is quarantine, disease control, disease, what all the diseases are introduced, how it is controlled, how the diseases are introduced from one country to another country, what are the different types of quarantine is there, how many stations are there, and what is DPPQS, where it is located, what it actually do, what is who is the present chairman, and all those things like stations, along with the detection techniques, what are the different kinds of detection techniques are employed in quarantine, and all those things you concentrate on. And overall review about all the topics. On the 43rd day and 44th day, you just concentrate on overall review about all the topics that what you have covered so far. And the next one, then pheneromic plant parasites. It's a simple thing, but we can expect a significant amount of questions from this pheneromic plant parasite. Recall the previous year question papers. At least there will be 1 or 2 questions in any way, some way. We have at least 1 or 2 questions from this section. So just concentrate on clear clause. Clause means Cuscuta, Loranthus, Verbank and Striga. And other important abiotic disease like disease caused by zinc, like which is not the factors involved in the pathogens, like no pathogens are involved, might be the nutritional disorders or maybe the some other environmental condition, we generally call abiotic, which means the biotic agents are not involved. In such a way you can do it. And the next one is 45th and 6th day, you can concentrate on important concept and exam related discussions, especially on biocontrol agents. In biocontrol agents also we can expect significant amount of questions, especially trichoderma, methods, mode of actions, some kind of things will be there, like mycoherbicides and all those things will be there. And we can also expect a significant amount of questions from this biocontrol, at least 2 to 3 questions. As I told you, putting small aspects all puts together, that will be very big, right? So just put all the small areas all puts together so that you can get a significant amount of questions. That will be helpful to get the net out, right? So 47th and 48th they concentrate on mushrooms, nematology and seed pathology. I gave just 2 days, so you must put a little bit extra effort and time to finish this. And 49th, the physiological and molecular pathology, DNA barcoding, actually not DN barcoding, it's a DNA barcoding, and hypersensitivity, illicit or phenolic compound, phytolactin, PR proteins along with the various types, along with the examples, it's important. On the 15th day we can concentrate on toxins, enzymes, growth regulators and polysaccharides that is produced by the pathogens for pathogenesis and their mode of actions along with. And 51, we will concentrate on hypovirulence, R gene, AVR gene, PAMP, DAMP, PTA, ETA, PRR, QTA, RNAi. Not me, you need to concentrate. I'm just giving an idea. I'm sorry for that. I'm just giving you an idea. Anyway, I will train you if you want, no problem at all. And you concentrate on, like, these are all the different areas. Since what are all the things we need to consider or concentrate on, that's why I'm just giving some things, right? Okay. And 52, laboratory techniques and methodology. And the 53rd day also concentrate on laboratory technique and methodology. So what are all the things we concentrate? Glass weights and the equipment uses, along with couple of laboratory equipments, microscopes and different types of microscope, their magnification power, who invented it, what are all the uses and all those things. And preservation and maintenance of the samples, what is agar and incubation methods, composition of agars and other things, sterilization methods and chemicals, blotting techniques. Are there 3 blotting techniques: RNA, DNA, protein, I think southern blotting, northern blotting, and western blotting, protein DNR and something else like us. Yeah. Electrophoresis, their methods, types, PCR and types, LAMP, ELISA, molecular technique, other molecular techniques on the fifth year. Probably it will take 50 to 60 days to finish the entire preparation. So at least you must prepare 2 to 3 hours, at least 2 to 3 hours, or else I can also suggest you, if you are a beginner, you know nothing about the net and all the stuff, I suggest you to go spend a little bit more time. I cannot tell you what exact amount of time you can put it on. Some people will get all the points maybe half an hour or maybe 1 hour, or maybe it depends on the people to person to person, because the amount of concentration that is doing. It's not about how much time you are studying. It's about how concentratedly you are utilizing your time. That is the important thing. Other aspects in exam point of view on the revision on the next days, I don't know how many days will left, but on the next days you keep on concentrating and keep on concentrating on the revision and all the other kinds of things. This is about what are all the things that we can keep it in mind. Okay. The method of preparation. Then many people used to ask how to prepare. What is the method of preparation? So this might be not feasible with everyone, but with the experience that what I have sorted out couple of points that is mostly useful for everyone. So the first thing is make your own note. That is a very important point. So you guys used to study lots of things in the lots of books, right? So again at the end of the day, or again at the just 1 or 2 days before the exam, if you want to recall all the books, that will be very difficult. So that make your own note. You can study the books that you want, you can recall the things, you can get the things, or you can get the information wherever you want, but put every single thing on your own note. I will tell you why you need to put your own note, because at the end or before, just before the exam, you cannot recall all the books because you might have forgot where actually you got the information. But if you have the book, you can recall it very easily. That's why I'm telling, have your own note. If you are studying some important thing or some important fact, write it down. Just go through once again, write it down, and I suggest you to select the best books possible, not every single book that is available in the market. Select some good books. Okay, make your own note. That is a very important one. Put a big note like 300 pages or 400 papers, whatever you are studying, you can make your own note. You don't need to write the things very long or big kind of things. You can tune it in a way so that your brain can catch it very faster, like hints or short kind of things, because the writing styles will be different. But I suggest you take a hints. Don't write like stories or like very big points, but just like a hints. That is a very important thing. Make your own note. That is the first point. The second point, taking hints are important points. Just now I told you, like not necessarily needed to write all the points, but taking the important points, like preparing the tabular columns, so that will be very easy to gather the huge amount of information in a very short table format, right? So that is also very important one. And the next one is underlining the fact from the books, that helpful for to recall, or if you want to again to get the book and see the information where you have studied. So if you have an important point, underline it. I suggest you underline with the pencil. You no need to go with some kind of pen or maybe stickers and so many kind of things people used to use, but I prefer underline with the pencil, because that is a book. Use like it a book. After 5 years or 10 years, if you want to give it to someone else, you have colored the entire book by using some kind of sketches, all the blah blah blah, it will be not, it not be useful, and it might not be convenient for others to read. So of course it is not necessary others to read your book, that is secondary, but to make a easy understanding and easy note on that, I suggest you to underline the important facts or important points that you are studying, and I also suggest you to make your own note from that point, because in the last minute we are not going to take all the books out and read the things. We are going to take our own note and just recalling the things. Okay, that is a very important thing. The next thing, have an idea about the previous year net question papers. Like we have published around the last 10 years net question paper. If you are interested, you can take a look into, or else you can also search somewhere else. It not necessarily needed to see our YouTube only, but you can also search other YouTubes, or you can also see other kind of things. But we have published the last 10 years net question paper with a separate title of playlist of net previous year net question paper. It starts with the 2006, 9, 2014, 14, 1, 14, 2, 18, 1, 18, 2, 19, 21, 23. Up to the last year, we have published almost 10 plus year net question papers. Why we need to go to the net question papers of previous years? To just have an idea on which area they are concentrating. And the previous slides have given the lots of areas where we need to study, how we need to study, around 50 days or 55 days preparation I have given, right? So that you will get an idea, right, where exactly they are concentrating. For example, methods of entry of plant pathogens. They are asking one question. So methods of entry of plant pathogens, like mode of entry, like, so at least there will be a question that is important one. More flagella types. So from that area, one question will come from. Like pheneromic plant parasite, at least 1 to 2 questions will come from. The, you can just take a look into the area where they are asking questions, but it is not 100% sure, like 100% sure that every year you will get the same area questions from the same areas. Of course, it may be different, I mean differs, but I suggest you so having an idea about the various areas where they are concentrating, that will be definitely useful for us for the preparation. Not only for the preparation, once you get the net done, the juniors will come and ask you, sir, how to read, Anna, how to read, or Akka, how to read, all those things they will ask you, right? If you want to tell them, you must have an idea, right? So that's why I told you it's not kind of coaching, it's a training you to become a master, training you to become a doctor. You will be the master of your own journey. So we will train you in that way. Okay, anyway, having idea about net of previous year, so that you will have an idea about which areas you need to concentrate. So these are all the things that you need to keep it in mind for the preparation. Of course, other points also may be there, but if you tell me if you ask me the one point we need to concentrate, I will suggest you to go with make your own note. That is a very very important point. Don't think about the net. It will be useful not only for now, but also for your future, and as well also for your juniors and your upcoming young plant pathologists. So make your own note. That is the best point. Don't worry about the time. Spend a little bit amount of extra time, since the it's approaching very fast. Maybe as I told you, in the second week or third week, maybe the notification will come. I'm not sure, but we're expecting. So make your own note. That is the best point. The next one, how to read the books. And in the market, as I told you, there are significant amount of, especially in pathology and agronomy and other kinds of things, there are a number of books are available to confuse us. The more options we have, the more confusion we have. The less options we have, the less confusion we have. That is the only problem. But anyway, don't worry, I have sorted out some couple of good kind of books, and you can ask me why not other books. See, you can just you do one thing. You just conduct some kind of test, like you will get 10 books from the market. You just go through with the standard books, whatever which were the information were there in the standard books, that was repeatedly returned by other kinds of books, are like if one information was there in the book, in this other book the same information was which was written in a different way, but the information remains the same. We must understand that. So it's not necessarily to read 100 books or 20 books or 50 books. Get some couple of very good books, 4 or 5 books, that's more than enough. Line by line, every single thing, not line by line, I would say only exam point of view. Since you are preparing for net, you are not preparing for ARS, right? ARS is a different thing, like preparation is different. So in such cases we need to go through line by line to have a broad spectrum knowledge, because viva also, interview also will be there, right? That's why. So anyway, since we are preparing for net, exam point of view, study the things and underline the things and make it your own note. As I said you earlier, it's not necessarily to read every single thing which were there in the books. A couple of things you may exclude it out completely, you can reject it out, no problem at all. And which are the things that we are not concentrating on too much. For example, disease of crops, that doesn't mean there is not plenty of questions are coming from disease of crops. Of course we have some kind of, you don't need to, what I'm trying to tell is, for disease of crops you don't need to study crop by crop, disease by disease in such a way, right? So that doesn't means, of course we might have a general basic knowledge about this disease of crops, all the crops, maybe horticulture and field crops, or maybe plantation, vegetables, various kinds of things. So having that broad general knowledge, you keep on updating. Sir, you can also, if you want, you can also reject that, but it's my point of view is not telling you that you can exclude that. What I'm telling you is the amount of questions are a little bit less when compared to the general kind of things where we are expecting. Anyway, at least maybe 5-10 questions also might be there, but if you're having a general knowledge on this is of crops, that will be more than sufficient. That's why I told you it's not necessarily to read every single thing. So you can exclude some of things by having the knowledge on the previous net question papers, which of the area that they are not concentrating, you can exclude it, no problem. And examples are very important. So every single thing whatever you are reading, examples are very important thing. We can concentrate on the example, so that sometimes, so mostly examples the probability of examples or asking examples are the chances are high. So that's a very very important one. And as I told you, underline the points. I didn't note it down on the notes that I have already told you. And as I told you, already mentioned in the history, one common thing that I have observed is every single year there is at least 5 to 10 questions. So history we have a very less, most probably maybe 10-15 pages. So if you read the 10-15 pages standardly, you will get 5 to 10 questions. What else we want, man? We will get easy answers. So we will concentrate on the history, and for reading the history you no need to go through the books. We have published the videos and we are going to publish the videos related especially history. We will cover the every single point what is utmost important from top to bottom. We will do every single thing. Watch the videos if you want, or else you can also get the information somewhere else, but you concentrate on this area, okay? And choose some important books are to finish it first. As I told you, there are plenty of n number of books are available. It's not necessarily to read all the books. Get some quality books, like important books. Like if you see the books, as I told you, conduct a survey, get the 10 to 15 books from the market or from the library, compare with the most standard books. Mostly the information was repeated from there to here, there to, okay? So in that cases, why we need to go with the 10-15 books, man? Just have some 3-4 good books and read carefully. That's enough. Like, so we can also save the time and we can also save the money as well, right? Now all books contains, as I told you, more or less similar kind of information only. So select the very few books, very few good books for a reference and as well as a preparation. So what are all the books to be preferred? The next commonly asked question is, I will tell you that these books are commonly available on internet. There, I am not telling the complicated books. Everyone might have told the same books. I am telling the same. A Plant Pathology, Alice and Jayalakshmi, Dubey, Mycology. For mycology, you can use Dubey. That's a very nice book, but a little bit extensive information was there. But still, it's okay, a nice book. And you can also select one more book called Introductory Mycology that was written by actually forgot Mehrotra and one more man actually forgot. Anyway, KR Anij and Mehrotra. That is also very nice book. That is also very nice book. Introductory Mycology. Probably third or fourth edition is running out now. That is also very good book for mycology. KK Mondal Bacteriology. That is very simple and very nice book. Go through it. And Kotle and Tiwari Elements of Virology. And one more book is there regarding RS Biswas, I think so, related to virology. Manoj Kumar Yadav for Plant Pathology. Kashyap and Man for Plant Pathology. These are all the common kinds of books that can be referred on the online. Then you can ask me. There is one more book. Very important book. A Vision into Plant Pathology, which was written by myself. You can ask me, you only wrote the book, that's why you are telling your book. You just go through the book, compare with the books available in the market, you will come to know. I have referred plenty number of books. As I told you, n number of books are available in the market. All the n number of books was I have gone through, and I have come up with a best source of information that is possible. This book, you can ask me, this book alone is sufficient? I would say the chances of passing the net is 70 to 80%, sometimes 90% if you read my book. That is A Vision into Plant Pathology, a complete student version which was published in the Kalyani Publishers. As I told you, it is not just because I wrote it. I know how much time I have invested. I wrote it for 5 years. I started when I was in the masters. I finished it just before finishing my PhD. 5 years I have spent on the book. Many people are writing books. Of course, there is nothing to say about it. And every single book is a really nice book. But I prepared with an intention of to produce a best book that is for the student. That's why I named it as a complete student version. Every single word have a meaning on it. A vision into the plant pathology. Have a student vision. How a student will think. In that way, I have prepared this book. Definitely, this book will be helpful. Take a look into it. Actually available in the Kalyani Publishers. If you don't wish to buy my book, of course, you can also read the other books. That's really good and nice. I'm just telling this book is useful, nothing else. Okay, hope you got the point. And anyway, so repeatedly asked question is JN Agrios book is necessary preparing alone for net? Preparing net alone, only you are preparing net. Is this JN Agrios book is necessary? I would say many people may not okay with my decision what I'm going to tell now, but this is with my own experience. For net alone, it is not necessarily needed to go with JN Agrio's book. I'm not telling another way. For preparing ERS to having the broad spectrum knowledge, I will definitely suggest you 5th and 6th edition of JN Agrios. Of course, 5th edition was written by JN Agrios. 6th edition was edited by someone else. Of course, that same information edited by someone else. Both books are really fantastic. There is no doubt about it. This is absolutely masterpiece. This book we can consider as Bible, Bhagavad Gita, Quran, whatever you want to consider. But preparing net alone, many people are asking. So for preparing net alone, is JN Agrios book is important? No. I would say why? Because you can see the books that is available in the market. Majority of the books, even book like me, what I even envision into Plant Pathology, we have took the lots of point, we have abstracted the lots of point from the JN Agrios book. So if you see in a common kind of books like us, 3 or 4 books, indirectly you are reading the JN Agrios book only. Why you need to go to again JN Agrios book? And you can ask me one more thing. Everyone are telling JN Agrios book. The perceptions is different. There is nothing else. Why I am not telling no need to for net alone JN Agrios is not important, because as I told you the first point, many of the books what you are reading, they got the information from the JN Agrios, which means indirectly you are studying the JN Agrios. Second important point, the people who are preparing for their first time or maybe already wrote for 1 or 2 times but they couldn't able to pass. Many people are telling like JN Agrios are important, but if you open the book is very vast. Like the 5th edition is around 922 pages, 6th edition is around 858 pages. It will be very difficult. By seeing the book itself, people will scared and run off, right? So to get out of it, Agrios book is not necessary for preparing net alone. Net alone, JN Agrios book is not necessary, because majority of the books, whatever we are reading, whatever you are seeing, all are directly or indirectly they drawn the information from the JN Agrios only, which means you are indirectly studying the JN Agrios, nothing to worry about it. For ARS, you can ask me, for ARS, is JN Agrios book is necessary? Absolutely yes. I would say 100% yes. For ARS preparation is different from net preparation. JRF preparation is different from ARS and net, and SRF is also. The preparation module and the kind will be different from exam to exam. For net alone, I would say no to JN Agrios, because I have told you the reason why. For ARS, I will definitely support the JN Agrios book, as I told you the reason, right? The next one, recalling or revision. So is recalling is that much necessary? Is recalling is necessary during or before the preparation? I will tell you whether it is yes or no. The first thing is revise all the things before you are going to exam is a very important. So while before going exam, you must recall the things. So if you don't have a time to recall the things, I suggest you to at least watch the videos of YouTube. We have published lots of videos previously, or you can also watch on the other YouTube channels. No problem at all. But ultimately, we need to get the things done, right? The second thing is the last minute preparation, how to do the things right. In the last minute, like just 1 week away from the preparation or just 5 days away from the preparation, no need to read all this theory kind of information. Go with the MCQ kind, because you are in the last schedule, because you are in the last minute. It doesn't work if you study theoretically. So that in the last 1 week, I will prepare you to read only MCQs book. In the last week, I am telling, revise your notes. That what if you have taken the points from day 1 to now. That's why in the last minute preparation, the notes that you are taking, that will be helpful, because you cannot read all the books in the last minute. So that the notes that you are taking, that will be helpful, first thing. And the second thing, read the MCQs book. That you can ask me, what are the MCQs book that we need to go through? That I will tell you. They have prepared some couple of important MCQs book that are available in the market. That doesn't mean the remaining books are not good. My intention is to sort it out the best book that is possible for the preparation, not only for net, other kinds of. Since we are working on net, let's talk about the net. Okay, and remember, revision before exam is a kind of meditation on knowledge. However, we are freeing our brain through meditation. So revision is a meditation on knowledge, where each review chapter that you have studied earlier, that actually deepens the understanding and the transformation of your all the things that you had studied, that converts into the wisdom. So the revision is very important. It's a kind of meditation. I suggest you to revise without don't tell that I don't have time, the last minute, all the stuff. Never ever tell. Revision is a very important one. So that's why I told you keep the note. Now you got the point why I told you note down all the points. This is the reason. Okay, Regarding previous year net question papers, so as I told you in the last 1 week, I told you to only read the MCQs questions, right? At the time you can see the net questions, or else I can also suggest you go through the previous year net question papers in the day one itself, so that you can have an idea where exactly the area they are concentrating. That will be useful. First thing, so what will we get by seeing the previous year net question papers? We will get an idea, right? So the geeky researcher, we have published all the last 10 years previous net question papers. That will be helpful for the preparation. The second thing, understanding the common area where they are asking the questions. By searching the previous year question papers, we will get a common understanding which area they are asking, right? And repeatedly asked questions also will be there in the net. You can see there are a couple of questions which were they're asking repeatedly. Every single the question will be there. So such kind of questions also will be there, so that we will have an idea, overall idea. To have an overall idea and the mentality of the pattern of net question paper, will get a little bit idea, so that will be helpful for our preparation, right? What to do in the last 1 week before exam? I told you just now, in the last 1 week exam, close all your theoretical books, put it on the cupboard, open the MCQs books, go through the MCQ book. No theoretical at the last 1 week. I would say last 10 days also, but 1 week is more than enough. One day you can finish one book, 7 days, 7 books, more than enough, right? And you can ask me, sir, can we finish one book in a single day? We are in a hyper situation. You must finish it, man. If the situation demands, you must do it. If the assignment last day is coming very fast, what we will do in what we do in our MS and PhD, we have finished it in 1 day, 2 day. We have finished lots of complex kind of assignments. Anyway, you guys also might have finished it. So in the last 1 week, it's a time you must do it, because you cannot say that I am a very slow reader, I can read very slowly, or some, such a way. You need to spend a lot amount of time, right? So since it is the last minute, you can just open the only MCQs book at the just 1 week before the exam, go through the facts fast and finish the book. 7 days, 7 books, right? The list of the some books I prefer, that doesn't mean the remaining books are not good. I am giving couple of books that is a very good books. I have read many books which are there in the market. Of course, every single book is a really fantastic book, I cannot say no on that. But by having a broad spectrum knowledge, or broad spectrum overall idea, I have prepared a couple of books for the last minute MCQs books. Okay, the first one is, I suggest you my book only, A Vision into Plant Pathology complete student version, because I told you I have gone through all the books, I prepared it as a complete student version. That's why first I prefer my book. The second one is, there is a book called Foundations in Plant Pathology, which was written by Bhupendra Singh Karyat. It is also very nice book. I have gone through this book. Maybe a couple of mistakes also there. Of course, in my book also there are a couple of mistakes. I have sorted out. Anyway, we will rectify it in the second edition. But this book is a very nice book, and this Bhupendra Singh Karyat is a very nice person. I have spoke to this person many times. He's a very nice man, very enthusiastic person, very kind and very good person. Anyway, not. Is a good person or something like that, but the book is very really nice. Since he's a good person, automatically the book will be nice, right? Anyway, so that was a really very nice book that was also published in Kalyan Publishers. Just take a look into. And the second one is most commonly mentioned book, A Plant Pathology Complete Version, which was written by Kashyap, Man and Kang. This is also very nice book. Still there are many mistakes are left, but still it is one of the masterpiece. The next one is everyone's favorite, Plant Pathology for Competitive Vision, Manoj Kumar Yadav. I spoke to Manoj sir also, and he's also a very nice person. He gave lots of ideas when I was writing my book, like how to publish and what are all the things that we need to consider when we are publishing. And he will speak very nicely. And I, not just because he is a nice person, as I told you, if you are nice, automatically the book will be nice. This is definitely a masterpiece and very nice book that was written by Manoj Kumar Yadav. And one more book, Objective Plant Pathology, that was written by Sushanta Banik sir, and I spoke to Sushanta Banik sir also, and he have given lots of good ideas for publishing things. And in fact, he also offered me, like, I will help you in proofreading, and in fact he told you many things, like how to do the book and how to change like this and like all those things. Definitely, if I get a time, I will definitely collaborate in the future project with Sushanta sir. And definitely this book is a very nice book. As I told you, if you are nice, automatically the book will be nice. Anyway, secondary, the book is a really nice book, very small book, but still it is a very important book and a very nice book as well. And Plant Pathology at a Glance, this book, Utpal Kumar Bhattacharya, and this is one of the most commonly repeated book, right? This is also one of the very nice and famous book that is especially meant for preparing net exams. And apart from all, I suggest my book. That doesn't mean other books are not important. You are in the last minute, so at least you select 5 to 6 MCQs books. These 5-6 books will be helpful. What are all they? I told you: A Vision into Plant Pathology, Kasyap Pandman, Bhupendra Singh Karyat, Utpal Kumar Bhattacharya, Manoj Kumar Yadav. All the books you just go through, take a look into. That books will be helpful for you. Nothing to worry about other books, because all are directly or indirectly having the information from GN Agrios, or more or less similar kind of information will be there in the books. Nothing to worry about it. You just concentrate on these areas or these books. Okay. And doubts, regarding doubts, how to clarify the doubts. Maybe see, whatever I have spoke from the last 50 minutes or 1 hour, still while you are preparing in the initial time, you may get lots of doubts, or sometimes your confidence will be low, sometimes you are not feeling good, or sometimes maybe someone are discouraging, some people are telling this is not good thing, or this is not the good book that you are reading, go with the other books, go with the other things, this book is important, Ginagri's book is very important for net, without the book you cannot get it, like that. So many kinds of things will come to your mind. There lots of people will come to confuse you, lots of people will try to put you in pressure. I cannot say I cannot blame on the person that they are confusing you, because they also have a different perception and ideologies. If you have such kind of doubts and you're not feeling well, and like you don't know where to start, where to go, and all those things, don't worry about it. You can contact us through mailing us in the Geeky Researcher, or you can also mail us through researchergeeky@gmail.com, or else you can find yourself, like you just enroll yourself in the Geeky Researcher website, www.geekyresearcher.com, by giving your name, phone number, where are you from, what are you studying, what is your aim, what you want to become, and all those things. We will help you out. Nothing to worry about it. Any doubts, all the time we will help you, since the net is approaching very fast. Until the net done, we will be in touch with you, and if you guys have lots of doubts and other kinds of things, don't hesitate to reach us. Okay, we will help you, no problem at all. Net thing, last thing, but the first thing, coming to the first point again, how to prepare yourself. Now you might have an idea by having all the points. Now you might have an idea, right, how to prepare yourself. You will have an idea what books you need to refer. You have an idea where to start. You have an idea, you put a separate notes and take the points. You have an idea like what are the books, the most important books that we need to consider. You have an idea what to prepare in the last 1 week, putting all the theoretical things out and bring the MCQs in. You have an idea like how to prepare, and nothing to worry about it, because every single book contains more or less similar kind of information. Get the good kind of books and start with it. You have an idea all the things how to prepare yourself. Now imagine yourself, you have already done the net. Nothing to worry about it. Everyone who are watching the video, or who are not also watching the videos, will get the net done this year. Don't worry about it. We will help you to our level best. And regarding the previously published videos, we have published lots of around some 15 to 20 videos on preparation of net also, and the links are which are we are giving in the description box regarding mycology, bacteria, all the things. Anyway, we suggest you to just take a look into, because we have mentioned the best information that is in the previously published videos. This year also we have prepared, but anyway, when compared to this year, last year, the information we have produced is absolutely masterpiece, because there is a reason for that I will tell you later. And we are giving the links in the description. If you are interested, just take a look into. And what's upcoming in the Geeky Researcher, what Geeky Researchers are doing this year. We have a plan, we have a master plan this year. What upcoming what Geeky Researcher upcoming with. We have a very good plan this year. We have prepared 50 plus net videos on preparation. Just not all kinds of preparation what you see outside, but we have prepared the quality and the best videos possible. Each video will last around 40 to 50 minutes. Around 50 videos, it will come more than 50 to 60 hours. Not just because hours, we have prepared lots of videos with very good good points, by considering important things, by considering the previous year aspect, by considering the previous important topics, all the things. We have prepared 50 plus net videos this year, which last more than 60 to 70 hours. That will be helpful for you guys to prepare this year net. The second thing, in the last minute preparation, we have prepared a video which lasts 5 hours 21 minutes. Just imagine. Then you can ask me, why this 5 hours 21 minutes, who will watch? See, this is the video where if you guys have no time, where the people are not getting the time to prepare from the day one or maybe in the first week onwards, in the last 1 week only you have a time, you don't have a time to prepare because you might be busy in research or might be busy in some other kinds of projects or other kinds of things. Those such a kind of people, we have prepared a last minute video which spanning around 5.5 hours. Trust me, this will be a mind-blowing video for everyone who are watching. Definitely it will be helpful for you. It's like a recap for all the things that we have done, and we have done in the recent last year and this year. Every single thing, the last minute recap will tell you about. And we'll, as I mentioned, we will clarify you the doubts and train you according to the level and the situation you are at, because some people, they are in the initial level, some people might be at the medium level, some people might wrote their net already 1 times or 2 times, but still they couldn't able to get the pass mark. Such kind of people with various levels, the existence of various levels will be there, right? So according to your level, according to your knowledge, we will help you how to prepare and all those things. The only thing you need to tell us is the truth, how much you know about the plant pathology. If you tell the truth, we will help you how to prepare. Nothing to worry about it. Why we need to tell you? You have the roadmap to prepare, right? You can prepare yourself. In the doubts clarification, you can reach us. Nothing to worry about it. And regarding apart from all the stuff, this is the book that I was written. It's been almost 2 years to now, and it is one of the best information that I have generated. I'm not telling just because I wrote it. Definitely, it will be a very good book for some reason. You will get it once you get into it. Anyway, that's secondary. You guys just take a look into if you are interested. A Vision into Plant Pathology complete student version. For further more doubts, clarification, many of you people might have a doubt how to reach a geeky researcher, how to contact them, all those things. We have a website called www.geekyresearcher.com. On the left side down, there is a query section. As I told you, enroll yourself. How to enroll? Very simple. Put your name, your address, what you're studying, what you want to become. What you want to become in the sense like, not like collector or something else, just jokes apart. Anyway, like what is your aim? You are preparing for NET or maybe ARS. ARS, this is the not the videos especially meant for ARS. This is only meant for JRF, especially NET. To be frank, especially NET. For JRF is different, SRF is also different, and ARS is completely different. The pattern is different, the mode of preparation is different. Anyway, we are preparing for NET since this is only for NET videos only. And you guys can reach us at www.geekyresearcher.com. You just mail us like researchergeeky@gmail.com, so that you will get our number. We will we will help you like in a different way, or else you can also mail us, we will reply you there, no problem at all. And this is about this year preparation. Definitely I hope everyone they are extensively preparing for this year. No need to worry about it, we will help you. We all sit together and work the stuff together, and I hope everyone will get this year NET done. Anyway, I wish you very all the best to each and everyone. From this onwards, we are going to publish the videos. As I told you, 50+ videos are on the way. The videos will come, and the last minute preparation videos will come in between. We can also clear you the doubts as well. No need to worry at all. And I wish you very all the best to each and everyone. Stay geeky and stay tuned. We are Team Geeky Researchers.